Okay. Hello everybody. Today we will continue to actually go through where we left off for transport implants. So in our lesson we actually talk about how is the phloem adapted for its function, where the phloem sieve tube elements have very little protoplasm, which are your nucleus, your cytoplasm and cell membrane, and they are generally actually there to help to reduce the resistance to the flow of substances within the phloem. Pores within the sieve plates will help to allow rapid flow of the manufactured food substances. In terms of adaptation, you have your companion cells that have numerous mitochondria to help produce energy through the process of respiration for the loading of sugar into the phloem sieve tubes. And every phloem sieve tube cells would have an associated companion cell to help to ensure its survival. So generally, these are how the vascular bundles are organized, and you could see that the xylem is actually located on the inner side of the vascular bundles, which is over here. And what happens is it separate the xylem and the phloem. There is a cambion. So the cambion actually helps to ensure that the cells can actually differentiate, divided, and together they will actually form the whole vascular bundle. In the middle of the vascular bundle, right, in the middle of the stem is actually your pith. So this is another thing to take note of, and that the stem is usually covered with this layer of cells called the epidermis, and these cells are usually protected with a waxy waterproof cuticle that helps to reduce the evaporation of water. This is one mechanism to help the plants from preventing the plants to undergo withering due to the multiple loss of the water. Okay, and then of course you have the cortex, and this is the region between the vascular bundle, and you could see that it usually exists together with the phloem area of the vascular bundle. So what happens is if the cortex is removed, the loading of sugar will not be able to happen. However, the plant can still actually transport water up and down due to the presence of the xylem. So generally, you will need to know the different arrangement and how the vascular tissues are actually organized in stems. Next, we look at the vascular tissues organized in roots. In the dicotyledon roots, there is actually the xylem and phloem, but they are not actually bundled together. They are separate structures. From the red color item, you could see that that is the area of where the xylem is, whereas the one in purple is actually the phloem. So how I remember is X marks the spot for xylem because xylem is starting with an X, and therefore the phloem is basically the the one that is within the stem within the roots that actually obtain the sugar and other minerals. Okay, another thing to take note is the roots, right, actually have all these very long structure, and this long structure basically your root hair cells. So if you remember in the chapter on cells, the root hair cell has a long and narrow protrusion to help to increase the surface area to volume ratio for us to actually absorb all the water and mineral salts. Okay, so the cortex of the roots is mainly a storage tissue, and the inner part is the endodermis. So the epidermis of the root is usually the outer layer of the cell, and this is a piliferous layer. And in this case, right, each of the root hair is actually just an outgrowth of an epidermal cell. And this is mainly to increase the surface area to volume ratio in order to absorb water and mineral salt. So you could see that this is how the vascular bundle is organized in roots. Okay. So in a nutshell, right, you have to actually know the positioning of the xylem and the phloem in three main areas: the leaf, the stem, and the roots. So the leaf, the stem, okay, and of course the roots, which is something like this. But of course I didn't draw I didn't draw the external elongated part of the root hair cells. So the ones that are highlighted in yellow, these are actually your xylem. Okay, so these are the xylem area, the inner area, and the ones that are highlighted in red are your phloem. Okay, this part here, the phloem will be the outer part, and of course these areas are your phloem. So in exam, these are the common things that can actually be tested for you in terms of identifying. It can come out as an MCQ. Okay, now how do we study the movement of substance in plants then? So first of all, we need to understand how substance actually move in plants. It is by the process translocation, where translocation is the transport of manufactured food substances like sucrose and amino acids in plants. What is meant by bidirectional, right? Bidirectional means it moves to the top of the plant and it moves to the bottom of the plant, and that food substances can actually be transported through the phloem tissue from the shoots and to the roots or vice versa. Okay, and what happens is to understand how the transport and movement of the translocation, we actually do a series of experiments called translocation studies, and these are the three main ones that you need to know: the aphyte, the ringing experiment, and the radioactive carbon isotopes. So these three experiments, right, came in this order to understand, to actually build a story of understanding the positioning of the phloem tissues. So for the aphytes, right, when we use the aphytes, right, the aphytes actually has a very long, the aphytes actually has a very long pith, a feeding stem, in which, right, what it does is it will actually penetrate through the stem, but it's not long enough to actually absorb the water. So what happens is the air fight, right, will first be actually antagonized with carbon dioxide while it is feeding on the stem, in which you cut off, and then after that you cut off its body, such that the proboscis actually remain in the plant tissue. Subsequently, what you also realize is that there is actually a liquid that is oozing out from the cut end of the proboscis. This is actually the area of the mouthpiece of the aphyte, which is the one highlighted in yellow. And because of it penetrating the phloem of the stem, as you can remember, the phloem is at the outermost layer of the stem. When we examine it, we will actually and test it, we will actually see the like reducing sugar present in the in the in the mouthpiece. So why is it necessary to anesthetize the aphyte while it is feeding? This helps to ensure that the body of the aphyte will be cut off cleanly when the aphyte is still feeding, to ensure that the proboscis will still remain the phloem sieve tube. Because if you were to trap it, it's alive, it can actually attach it out, and then you will never know what kind of solution, which vessel is being punctured. So when we do the food test, right, we can use Benedict's test to test for reducing sugar, Burette test to test for proteins, and ethanol emulsion test for fats. So these are the ones to analyze the solution. So what we found is that the A5 actually contains a lot of reducing sugar, and through this, right, we are able to identify that the outermost vessel may contain phloem, or it's mainly a, a vessel that actually is able to transport sugar. Okay. So next, to confirm that this is consistent throughout the whole circumference of the stem, we will use the ringing experiment, which is the second experiment for your translocation study. First off, you cut off a complete ring of the bud from the stem of a twig, and then you have B to act as the control. Later on, you use the ringing experiment, you set it up and ensure that the bottom of the tweets are both in contact with water. This is so as to allow the plants to actually take up water. And what will happen is water can still be transported up. However, the phloem is no longer available. So while the plants, the top layer can still undergo, because of the presence of the leaf, it can undergo photosynthesis. You'll realize that the sugar for A will tend to accumulate at the top layer and it forms a bulge. Whereas for B, there is no such bulge form. And that is because, right, the removal of the phloem helps to prevent the translocation of the sugar to the region below the ring. And this accumulation of sugar above the ring will then lower the water potential in the region, allowing water to enter the region, resulting in the swelling. So what the experiment has actually shown is that the phloem is actually located on the outer circumference of the stem. Whereas the next thing is we want to double confirm the exact position. Is it a continuous one circumference or is it in different vessels on its own? That is still located on the circumference of the stem. So we actually use a radioactive carbon isotope. This radioactive carbon isotopes is carbon-14, and this is actually being put in together with oxygen to form carbon dioxide. So the carbon dioxide will actually carry the carbon-14 isotope. Now plants will require carbon dioxide for photosynthesis, and they will use the carbon from carbon dioxide to form glucose, okay, or even sucrose or starch during photosynthesis, and this will be transported along the phloem. So when we actually cut a section of the stem and expose it to an X-ray photographic film, you'll realize that the stain area would actually contain the carbon-14. And what will happen is this region where the radioisotopes will be detected, and that this will be detected in the phloem area. So that is how we actually identify through the process of where the exact location of the phloem, where the A5 experiments has established that the phloem is located at the external ring of the stem. The ringing experiment helps to determine that, to confirm that the A5 is not at just one particular point, but it's really at the external ring of the stem. And finally, the radioactive carbon isotope test helps to verify that the individual vessels that is found throughout the at the circumference is the one located, and it's not just one continuous vessel. So next, to study the pathway of water is very simple. You basically put a plant in colored dye, and then what happens is after a few hours you cut a transverse section of the stem and the roots, and you will slowly identify which of the area are being stained red. And this is where the ones in that are highlighted in red based on the root, stem, and the leaf are the areas where you will actually be able to see the solution being stained red. Why? Because xylem is the tissue that transports water in a plant. Okay, in our next lesson, we'll be talking more on the entry of water into a plant and how water is moving against gravity. Thank you.